Hello and welcome to Reasonology. Today is Sunday, the 28th of June 2020, and the big news is that the corona pandemic has crossed the giant figure of 10 million infections worldwide. The death toll is half a million so far, making the mortality 5%, which is higher than the estimated 2%. So today we are going to discuss one of the recently published report in the Wall Street Journal by the Center for Disease Control Korea (CDC), and they claim that the reinfection of already recovered COVID-19 patient is not possible. According to this report, these findings came after some shocking results of the 280 patients who were recently recovered from the COVID-19. They observed that the real-time PCR test of these patients was still positive even after weeks of recovery. Almost similar observations were reported in a study conducted in China, where stool samples from the already recovered COVID-19 patient were proved to be positive even after 28 days of recovery. So, how the corona reinfection is not possible? Let's Reasonology help you with some scientific logics to understand this claim. The respiratory epithelial cells, which has the ACE2 receptor for binding of the coronavirus, and most of these cells are usually infected by the coronavirus. These epithelial cells has a half-life of 3 months. So there is a big chance that the patient will be shedding RNA of the virus even after 2 to 3 months of recovery. Professor Schiffer, who is head of the Vaccine and Infectious Diseases Division at the Washington State University, also suspects that these small fragments of virus flare-up might be causing these positive tests in those patients which were previously displayed as negative. Most of the viruses which cause chronic infections, for example, the retroviruses like the HIV or the hepatitis B virus, they usually integrate its genome into the host cell and then start production of the new viruses. Unlike these viruses, the coronavirus remain in the cytoplasm, and that's why the coronavirus cannot cause the chronic infections. Also, when compared to the herpes and chickenpox virus, which remain dormant in the human body for years, it is quite unlikely for the coronavirus to settle in the human cell, lay dormant, and then resurface and cause infection. Recently, it's been observed that the genetic variation in the coronavirus is quite less compared to other RNA viruses. So, the memory cells in the human body can easily clear these new viruses with the production of macrophages and antibodies. These findings are quite encouraging with regard to the vaccine research for combating the COVID-19. Lesser the genetic variation in the genome means the vaccine will be more effective for longer time.